Good morning, everybody.
Good morning.
You are right now almost 1 o'clock in the science track room for fantastic yeasts and where to find them. Before we get started, there's a couple of ground rules I want to go over real quick. While you're here, please have your phones on vibrate or silent mode. That way we can have a good interaction between us and the panelists. Second, at all times, please have your mask over your nose and under your chin, covering both sides right there. That way we can prevent the spread of COVID. Though we're here, I'm going to pitch a couple of big panel that I'm supposed to real quick. Though this is fantastic yeast, we do have making weird creatures next. Sorry, words are hard this morning. And then our big panel tonight is science versus movies, which is going to be very fantastic. If you ever want to see scientists just hurt themselves trying to prove science right in movies, that's where you need to go. Lastly, I do want to say we do have some cool N95 masks right here if you want to get some after the panel. That's great. And we are promoting the Big Brothers Big Sisters charity of Metro Atlanta. So at the end of the panel, if you want to donate, great. It goes to a great cause. If not, that's fine. But every dollar that we raise, Dragonfly will match up to $100,000. So help us make them pay. Anyway,
Make them pay. So good morning, everybody. Welcome to Science Track. If this is your first visit to the track, welcome back to Science Track. If it is not, this panel is fantastic yeasts and where to find them. We have a trio of magnificently experienced panelists here before us, and I'd be happy to introduce them to you. Your leftmost panelist on the far end graduated with a bachelor's in microbiology from the University of Maryland and went on to earn her doctorate of dental surgery at Howard University in Washington, D.C. She's a passionate cosplayer known online as StitchesAndSparkles. Will you please welcome Dr. B. Huseman. Our middle panelist is a Master of Organic Chemistry and a Master Division costumer. She uses the same skill set in both accomplishments. When she isn't teaching as an adjunct at a Seattle area community college, she's dressing up as a superhero or a Star Wars character to bring joy to everyone and raise money for charity. Please welcome here Tori Stenmark. And immediately to my right is a woman who works as a postdoc in astronomy at Uppsala University, studying the atmospheres of extrasolar planets with the goal of finding potentially habitable planets in other solar systems. She also loves doing science outreach, especially with kids. Welcome, kids. doing outreach right here and now. When not working, she can often be found playing music, hiking, or teaching martial arts. Please welcome Jennifer Greco. My name is Jeff Hubbs. I'm a consultant working in the fields of energy systems transition and climate change. I have next to no biology background. So the feigned ignorance some panel moderators get to affect in a panel like this, in my case, is not feigned at all. It's genuine ignorance. So I'm just going to sort of pretend I'm Dick Cavett and just let these people have at. Although I promise I will not drop the name of a beetle into casual conversation. So let me start off by just asking the basic question, what even is a yeast?
So yeast are single-celled fungal organisms. The fungal kingdom is one of the big ones, and a lot of people don't realize that, well, maybe you guys do because you probably know a little more about science than your average American, that fungus are actually much more closely related to animals than to plants. Their cells are much more similar in structure to the animal cells, just with a chitin cell wall. So yeasts are the single-celled fungus that we love because they make all of the fabulous things in life.
Very good. So other than in our pantries and other places where we buy yeast as a product for some sort of use, the title of the track is "And Where to Find Them." Absent human intervention, where do you find these things in the wild?
Well, start.
I mean, if you're trying to make your own sourdough culture, that was everyone's big hobby. You just sort of see what's on the flour or in your kitchen. But that's still kind of human intervention because at some point you probably baked bread in your kitchen or someone else baked bread in your kitchen, and the yeast is probably not wild so much as, feral yeast that you can then recapture.
I mean, they've cultured yeast from air that they brought back down from NASA test pilots, so you can find yeast in space as well as on Earth.
And not exactly absent human interaction, but there's a lot of yeast that lives in and on you.
The, what is it? Saccharomyces cerevisiae. I'm not a biochemist, so I'm probably not saying that right.
That's how I say it.
Okay, you're a not a microbiologist. Technically, my undergrad degree says biochemistry, but that was a while ago.
Yeah.
So that's the species that we mostly put in our bread and stuff and our beer and stuff. But there's lots of other yeast species out there, some of which are more or less pleasant to have to deal with when they colonize humans. So.
Candida albicans is one of the big ones. It's what causes thrush and yeast infections. Yeah, real fun.
Don't put that one in your beer.
No.
But if you did, what would happen?
Just don't, okay? Your scientists were so busy trying to see if they could.
I realize as scientists you want to see if you can, but sometimes just don't.
Nuke it from orbit.
It's the only way to be sure.
I have a real question. So different beer
Can we use the mic for questions so
Yeah.
that everyone can hear?
Hi. So the different beer companies, do they distinguish beer by they have actually different yeasts, or it's exactly the same one?
As I understand it, it's a different strain of yeast.
Yeah, I think so.
It's like if you go get tea, all tea is pretty much if it's tea, it's all Camellia sinensis. But black tea and green tea and oolong all taste very different, and within that you can do a lot of variations. Yeast is the same thing. It's the same organism, but it's a different strain. It's got a different, maybe a slightly different combination of details and bred to do this, that, or the other thing. So lots of dogs are different, but they're all still dogs. It's that kind of thing.
Just as an open question, do beer companies, is yeast something they just buy as a commodity product from out on the free market, or is it something they cultivate themselves over time?
To my understanding, it's generally cultivated. So most yeast reproduces, for the most part, via asexual reproduction. So you're going to get pretty much clones of that same cell again and again and
Right.
again. And it only reproduces sexually under times of stress. So for the most part, your yeast is going to breed true. And so if you've been cultivating a certain yeast that gives you a certain flavor, certain qualities, you're going to want to keep that for consistency.
So remember I said they found yeast in air brought down by NASA test pilots. So there's a little bit of a story there that ends in beer and begins with the Apollo anniversary. So what happened is that in 2019, for the anniversary of Apollo 11, there was a Maine-based brewing company which has an excellent name. They're called Maniacal Yeast Labs. And they somehow got in touch with a couple NASA test pilots who were flying at 70,000 feet in the stratosphere, and they were like, hey, could you bring me back a petri dish full of air from the vents of your whatever they're flying in? And the NASA test pilots were like, sure. And so they brought back this petri dish full of air, which they then cultured to cultivate 3,500 strains of yeast. And then they picked one of these to make beer, which they released on July 20th, 2019, and it's called Interplanetary Ale.
That does kind of have a Andromeda strain ring to it, doesn't it?
I think you can buy it, yes. You can look up Interplanetary Ale and let me know.
So we had a question over here somewhere? Okay.
So when you have that many yeasts in your petri dish, how do you just how do you pick one out to say, let's try that to see if it tastes good? How do you tell whether it's going to be? Can you tell ahead of time whether it's going to be one that you just shouldn't do, or this is a good possibility? How do you break it down?
So I haven't done on any actual culturing of yeast, but in general, when you want to kind of separate out all of the organisms you've collected from an environmental sample, you need to do a streak plate, where you take like a tiny little bit of all of the gunk that's grown on it, and then you spread it out and you dilute it until you get all the different cells individually making their own little colonies. After that, then you can grow it up and test it on different plates, see what it wants to grow on, see what it's going to actually thrive in. But beyond that, as far as it's going to make good beer, you got to just try it.
So there is a little bit of humans are really good at detecting stuff that is bad for us. Not always, but like if you smell it and you go, that's probably a sign that there's something in there you shouldn't eat. We're really well attuned to, rotting milk, rotting fish or meat or stuff like that. We're very sensitive to it. So if you've got a yeast culture and it smells like I would say not that one. Sometimes sometimes it doesn't smell like that, but it's, and you're like, well, let's see. And you make a small batch of beer and hope it's okay. But people are surprisingly good about detecting things like that.
Well, is it the case that for beer making, the point isn't to leave the yeast in the final product. It's the yeast is there to process some stuff into other stuff, and maybe that doesn't so much depend on exactly what yeast it is. I don't know. I'm just wondering out loud.
Yes and no. So part of the nature of what yeast does is that it chews on various sugars, whatever you feed it, and it produces ethanol and carbon dioxide. In bread, you mostly want the CO2 for the lifting, the rising effect, and in beer, you want the ethanol and also the bubbles. And in some forms of, as you get into like wine, maybe you don't want it carbonated, but you're still keeping the ethanol. So that's, the first thing you care about is the ethanol and the CO2, but you also are going to, everything else makes a difference too, right? It's all in there. So it will still be some effect. If it literally all tasted exactly the same, then you'd only ever buy Fleischmann's yeast from the grocery store and just use that.
Go ahead.
What was the situation where yeast would reproduce sexually? Like what kind of environment would cause that to happen?
So generally stress, which can either be due to lack of resources, like starvation mode, or if they've been reproducing too many times and getting too many errors in their genetic code, then they can undergo sexual reproduction in order to kind of eliminate some of those deleterious errors and get back to a healthy genome.
So can you give it, maybe just an overview of where we use yeast in? So, beer and bread are the obvious ones, but, kombucha, kimchi, yogurt, which of these involve yeast? What do we use yeast for? Which things are other things?
Yogurt is not yeast, I'll say that.
Yeah.
That's just bacteria. But kombucha, vinegar, that's really a furthering of the winemaking process.
I bake a lot, so I use it mostly in making bread and stuff. And one of the interesting things, I moved abroad last year, and I used to have a sourdough in the U.S., and then I started one when I moved to Sweden, and my bread comes out a little different because, of course, it's different yeast. And I have a colleague who actually brought his sourdough from France to Sweden, and his bread is different than my Swedish sourdough is. And so it's been kind of an interesting learning experience for me, and just how different yeast from different countries makes different bread, which has been really cool.
Sourdough, or at least the sourdough culture I have, it's not just yeast, it's also bacteria. So it's Saccharomyces cerevisiae, which is the yeast, but also Lactobacillus sanfranciscus. San Francisco is, of course, famous for sourdough, and that's where that comes from. Yeah. So with a sourdough culture, you are very much playing with a combination of things, and the terroir of your sourdough, if you will, of your kitchen is an important aspect of that.
Yes.
So, hold on, I wrote this down. Yeast can perform both aerobic respiration and fermentation, but they usually use fermentation even though it's so much less efficient, like even if they have access to oxygen. Do you know why that is?
It's been like 8 years since my microbiology
Yeah.
class, so I'm not sure.
Yeah, nah. I remember I can see the diagrams from my biochem Raymond and Johnson textbook, but they're very, very fuzzy and very far away.
Yeah.
You know the answer?
So the theory is, the model is that the yeast is basically carving out its niche by pumping out alcohol that other things can't deal with. It keeps other things from living in the fruits or whatever its natural environment is. So yeast can handle a lot more alcohol than other things. So it pumps it out and basically poisons the water for everything else, and then it gets to have it all to itself.
Cool.
Interesting. If y'all couldn't hear that, the yeast makes ethanol, and ethanol poisons everything around it. That's really cool. I did not know that.
So when it comes to killing yeast in humans, we use the azole family, so like ketoconazole and metronidazole. I'm just wondering, like, how they found out about that and why they know azoles are the best, like, at killing yeast, and actually how do they kill the yeast?
I just write the words on a prescription pad.
I mean, drug development is a lot of trial and error. Like a lot of. Well, sometimes it's like, well, this randomly worked, and this kind of sort of worked, and like the story of discovering penicillin, which is an antibiotic, was Pasteur didn't do his dishes. No, excuse me, it was Fleming. It was Alexander Fleming didn't do his dishes, and like came back and was like, there's mold on all these, ew. And then wait, there's no bacteria around these molds. That's gotta be something. But you should still do your dishes, she says to herself to remind her to do the dishes.
I can only tell you that space radiation kills yeast, but I don't recommend that because it also kills humans. So please don't treat your yeast infection that way.
Going back to sourdough, how do you keep? I've tried to do sourdough. I've started my own. I have bought cultures and tried to keep them. They never last more than a week or two. What can I do to keep a good sourdough culture going?
Ooh, good question. First of all, I'll recommend the book that my friend gave me when she gave me my first sourdough starter. It's called Alaska Sourdough, and it's a great sort of guide on how not to screw up your sourdough. I mean, you guys have had longer sourdough cultures than I have, but like, I mean, I feed mine regularly, so I know you can feed it different things. I feed it 50% white flour, 50% water, and then I mix it so there's no lumps, because I was told if there's a lot of lumps in your flour, then it grows like bacteria you don't want. And then you make sure you feed it regularly. One of the things that I do, because I was a very, very busy grad student when I was starting my sourdough, and I did not have time to remember to feed it as often as you're supposed to, so I keep it in my fridge, which means that if I forget to feed it, it forgives me more than it would if I kept it on the counter. And that was very helpful when I was doing my PhD, and I only remember to feed my cat because he screams at me if I forget. I don't know. What can you add? I'm sure you know more than I do.
So I got really lucky with my sourdough culture. I am the third generation of college chemistry teachers to have it.
Which is awesome.
So this starter is 130 years old. So obviously, like, my bacteria are not that old in yeasts, but it's 130 years old, which came down, let me see if I can remember, came down from, it started in Mississippi and got handled down to Professor Harold Jones, who handed it down to Ted Lindemann, who was my undergrad mentor and just a wonderful, wonderful person. Love to blow things up, love to set things on fire, but would also every year, or every month or so, would do sourdough pancakes for the entire chemistry department. and like pretty much every month. And I was about to graduate, and I went, Ted, what am I going to do? Because I'm not going to be able to have pancakes anymore. And I was just being, sad and a little silly. And he immediately goes, I'm going to give you some starter. And I have to this day, it's in the exact jar he handed me with the same piece of tape on it. And so part of it is simply that I got lucky with a really good culture that lasts. But I keep it in the fridge, and I feed it. I try to remember to bake bread with it, and I do not bake as often as I should, but it lives in the fridge. And when I do, I feed it, same thing, half water, half water, half flour, and a little bit of milk, like powdered milk, to give it a little bit more sugar and stuff to chew on. And I think a lot of it is just getting lucky with a really good, resilient culture. And sometimes they just don't take. Particularly like if you're culturing from your kitchen, again, maybe you get good stuff and maybe you get wallpaper paste and it's green and weird. And in that case, throw it out and start over, or go ask your friend if you can leave a bowl of flour on their kitchen counter for a week and see if their kitchen grows better stuff. I find, and again, maybe it's just that I'm lucky, but I find it's a lot less precious than people say. Like, oh, feed it every single week. I'm like, are you kidding? I wish I could bake bread every week. It's more like every few months, and it still survives. So finding a good culture, I think, is the important part. I am happy to share if folks email me after the panel. If you take the time to reach out to me, I will mail you some sourdough. Like it ships pretty well. But I'm going to make you do the work. I'm not going to just randomly send it, so you have to ask me for it.
I was just about to say, the very first time I had sourdough, I was in college, and my friend's mom lived in, we were in Southern California, her mom lived in Northern California, and we're like, oh, we want to start a sourdough culture, and her mom's like, oh, I'm going to put some in the mail, and I'm like, what? And so I get this package, the strangest package I've ever received in my life. So it's a plastic padded envelope, and I open it, it smelled kind of, weird, and I open it up, and there's like several plastic bags worth of like a cup of sourdough starter in it, and that was how we got it was how I got San Francisco sourdough in Los Angeles and later in Ohio, was by mailing it. And so I recommend that, especially if you're starting your first sourdough culture, to get starter from a friend, because also getting starter from a friend comes with a friend who can give you advice. Like I would call her and I would be like, it looks weird today and I don't know what I did. And then we could talk about it and she could give me advice about what she feeds her sourdough and give me recipes. And that was really useful. My brother once called me at 11 o'clock at night because someone dropped sourdough starter at his house and he didn't know what to do with it. So use your friends as resources as well if you know anyone that has one, or look online. There's a lot of good resources there.
Is this like the zucchini problem? For those of you who don't know, zucchini overproduce like nobody's business. Yeah. And in parts of the Midwest and the West, make sure you lock your car at the end of summer. Otherwise, you'll come back to find the backseat full of zucchini. The car will still be there. It'll just be full of zooks.
Speaking of temperature, what does temperature do with yeast? Does it stop growing below a certain temperature? Does freezing destroy it? Does heat that's not quite up to boiling destroy it? What kind of range does it survive in?
Depends on the yeast.
Yeah. I mean, you certainly can freeze it if you get those big Costco bulk bags of yeast, portion some out and just freeze it and then pull, thaw
You
it
don't
out what you
destroy
need
it to freeze it? Or
No.
not? Okay.
Well, it's if you get that dry yeast, that those little brown granules that are like, I don't know, bigger than a grain of sand. Yeah. Sporulated.
Okay.
So I do home brewing, and I've been considering trying to preserve the yeast when I'm done. If I find a strain of yeast I like keep using it. But based on what you were saying about yeast engaging in sexual reproduction when they're stressed, and that the entire point of home brewing is to basically stress them to the point where they become dormant again, am I running the risk that if I do this, say, 2 or 3 times, I'm just going to lose the character of the yeast as it was originally cultivated, because now it's exchanging genetic material instead of just cloning?
So it's still only exchanging genetic material with itself, pretty much. They tend to only sexually reproduce with other members of the same colony. So I would think you'd be pretty okay for that.
in brewing, different strains can affect the flavor profile of the end product. If yeast is just producing carbon dioxide and ethanol, how are the different strains affecting that flavor profile?
Well, it's not just producing carbon dioxide and ethanol. It's making a whole lot of other things as it goes through these pathways that all are going to add different flavors and change that overall taste.
I always think of it like the difference between the fancy vanilla extract that's like Madagascar vanilla or Tahitian vanilla versus the synthetic vanilla that's Fred Meyer branded, which is just vanillin and ethanol. Like, yeah, that vanillin is the major flavor ingredient of vanilla. But if you get the expensive stuff that's the extract of this strain of grown on this island or that island or whatever, it's dramatically different when you can taste it. And you're like, okay, yeah, no, I do see the difference. And sometimes, you know, you can just use the cheap stuff because that's all you need. But different types of yeast will have that same sort of thing. Like, everything else is different. Do you care? Do you not? What are you doing with it? Okay, I know this panel is fantastic yeasts. Can you talk about not fantastic yeasts? Are there killer yeasts? Like, what are the, like, bad yeasts out there?
Things that can hurt you.
It's a mold. There are a lot of them out there. Most of them are a little bit outside of my wheelhouse, but the good thing is that most of them won't affect you unless you're already immunocompromised. Our bodies are actually pretty good at fighting off most yeast, and even Candida albicans, the most common one to get a yeast infection from. Unless you have something going on, you're pretty much going to be safe from it. So that's why antibiotics can knock out the bacteria that usually are competing with the Candida, and that can result in a yeast infection.
Going back to the vanilla for a second, in the production of those beans, aren't the bean pods fermented to produce that? Do you know if that's a bacterial or a yeast-based system?
Good question.
I don't know off the top of my head. I would have to look into that.
Yeah.
Yeah, no, I don't know.
So I live in North Florida, and I am terrified of the what grows everywhere. I mean, it will grow on everything, mold, bacterial spores, everything you can think of. So I always get a little nervous leaving a bread culture out for any really period of time. And so can you speak to like the risk of like what you may need to cover if you decide to like have it on your table instead of like in your fridge, and what way you can protect it from the ambient yeast that's going to be out there, because some of that stuff is, if you've been to Florida, you know.
I know when I lived in Southern California in summer, we would all put our yeast cultures in the fridge because I one time accidentally ended up with a fruit fly farm instead of a sourdough culture.
Oh, yeah.
So don't do that. And so I would sometimes, especially during the hotter, more humid parts of the year, I would keep it in my fridge. And I kept kind of a loose lid on it. Like, I didn't seal it, but I keep it like in a plastic Tupperware with the lid sitting on the top so there's a little airflow to kind of try and keep at least more things out of it. It didn't stop the flies, but I think maybe it helps a little bit.
I usually grow my, when I do like the overnight, grow the sourdough to feed it and stuff. I take the stuff out of the fridge, dump it in the bowl with the flour and the water and the milk, cover it with a towel and stick it in the oven off. Like, usually what I'll do is gently warm the oven up to like, my oven has a warm setting, so I hit it to like 140 Fahrenheit, crack the door, let it cool a bit, and just stick it in the oven closed with a towel over it, and that seems to keep it reasonably secure. Now, I live in Seattle, which is not Florida, so the local ecosystem is going to be very different there. But I find that sort of a reasonably secure. It also means no one else is going to, accidentally clean it or mess with it or stuff, because it's in the oven, it's out of the way. You do have to warn your roommates so they don't try and bake cookies while you're culturing. Oh, no. That's never happened, but I always sort of like, let me just mention that there is bread or proto bread happening here.
I'll add, because I've lived for a number of years in places where it gets very cold in the winter, that the putting it in the oven method helps with the fact that with it rising better when it's really, really cold.
Keep that, like the ideal one is body temperature, right? Because the yeasts are meant to grow at 37 degrees Celsius, which is human body temperature. So you can keep it around that. Dr. Lindemann used to say, he sent me a file with my starter, and he said, on camping trips, I sleep with the stuff. So he'll have a sealed plastic bucket, and he'll put it in his sleeping bag and let his body keep it warm. And then you have sourdough pancakes for breakfast over the fire. His pancake recipe is just like to die for.
Hey, so putting on my mad scientist hat for a second, have there been any successful examples of any bioengineering or genetic tinkering done with yeast? And what were they done for, if so?
Oh, can I start? In prepping for this panel, I googled the word space yeast, which if you have free time, I highly recommend. It's really entertaining. And I think my favorite thing that I found is that there's a lab, and I forget where, that's cultivating a strain of yeast they found that lives on urine. And the reason they're doing this is so that when, like, astronauts go into space, they can make this yeast that feeds on urine and produces omega-3 and polyester. And so they're trying to see if they can make this yeast feed on waste and produce, like, plastic or nutrients as a waste disposal method, which I want this to work so bad because it would be really, really cool. And also, I'm just like, what?
Yeah, I was going to mention that if you're keeping your yeast in a mason jar, you can get airlocks that'll let the CO2 off gas, but keep air and other bad stuff from Florida getting in. So that's something you can look at online.
It's good to know.
I know there's a lot of other things other than bread. We've talked a lot about bread, but I'm about to start my adventure with mead. But that, like most people, you guys mentioned the pandemic pushed me into yeasty business, and sounds weird, but I remember when I first started out, I killed my starter like 3 times in a row. And I found out via YouTube that tap water, most of it has chlorine in it. And so it kills the yeast immediately, so it kind of rendered it useless. Did you guys find any other maybe people that are starting out in any form of brewing or baking or anything involving yeast? Like, what are the quick tips and things to avoid? I know we've touched on it a little bit, but just figured out.
If you're doing any kind of brewing, you want to start with really clean vessels. So like Star San, the best stuff, because it's going to be flavorless. It's not going to leave your stuff tasting like dish soap or anything like that. But very clean things, because you're making an environment where things want to grow. You're putting sugars in there. You're giving it a nice warm environment. And you want your yeast to be the one that's doing most of the work, most of the growing, not everything else.
Yeah, temperature control, and make sure it has food. If you bake with like commercial yeast, it'll always tell you proof the yeast first, right? Put it in with some warm water. Make sure that water isn't too warm, because you did ask, if you boil that water, it will kill your yeast. So human body temperature, pretend the yeast is a baby. Check the temperature against the inside of your wrist, because the skin's much thinner there, so check it on the inside of your wrist, and if it feels too hot, it's too hot for the yeast as well. Temperature control, give it a little bit of sugar. Sometimes you proof the yeast in the warm milk, because milk has a lot of sugars in it, or just add a couple pinches to get your yeast, give it the best possible start, the best possible environment it can grow in, and then turn it loose on everything else. So you want to raise it right, and then let it do its thing.
Can confirm, killed yeast with boiling water once. Not good bread. would have to look into this to be 100% certain. When I was given my sourdough, I was told to feed it white flour, not whole wheat flour, because if I fed it whole wheat flour, it would be too many nutrients, and then it would grow bacteria I did not want. And so I would say kind of be careful what you feed it, and I know you can feed it other things besides white flour because the person who gave my current starter to me was feeding it rye, but I would just say do some research on what you're feeding it to make sure if you want to feed it something different than plain white flour, you're not going to accidentally make yourself a bacteria culture you don't want.
I believe so. Chlorine in the tap water can be a problem. You can use, filtered or distilled water. I believe if you leave the water out in an open container long enough, most of the chlorine will end up evaporating out. So if you leave that, be careful about your environment. But if you can safely just leave an open, bowl full of water, that will mostly clear out before you put the yeast in. I believe you might want to do a little bit of research on that to be sure, but I'm pretty sure that works. Good to know. Thank you. The fish tank people say don't do that because the form of chlorine they're using now won't evaporate out. All right. Cool. Cool. Treat it with chemicals. I don't know about so treating your fish tank and treating your yeast tank is not the same thing.
Yeah.
So do some separate research. Yeast cells, B was right. Yeast cells are kind of like animal cells, but they're not animal cells. So
No. Please don't make fish yeast.
That- Yeah.
Ancient whale yeast.
I'm not sure whether I wanted to know that or not. If you missed that in the front, apparently they turned ancient whale yeast into beer.
So whenever I've been when I've been doing my brewing, always been told to put it into dark places to prevent light from getting in. Has there ever been a study done where they've done like light spectrum coloring to see if like a certain color could make it grow faster, better, provide different flavors, anything like that?
Yeast don't have chlorophyll or do photosynthesis, so I'm not sure. That might be more to do with like photodegradation of certain compounds.
So, sure. I once, so I took bioanalytical chemistry in college, and one of the things we did as a project, it was honestly more of a statistics project or an experiment design project, was let's set up an experiment where we change a bunch of different variables all at once, but you set up sort of a grid of conditions that you're changing such that you can do one battery of experiments and test a bunch of different sets of conditions. And what we did was growing kombucha on tea. And some of the tests we did was light versus dark, was growing them like some of them were underneath the lab sinks and some of them were up on top of the lab benches. I cannot remember what the differences were. I do remember, so the instructor said, okay, nobody has to taste it. But we were all in college and we all did, and like, wow, it was vile. Like, I respect that some people might like the taste of kombucha, and I've had some that's like, okay, but the lab kombucha was real not good, y'all. It was real bad. And I don't remember the specifics of whether the light made it better or worse. I think it was just kind of all worse and also worse.
Back to the space yeast. Yeast that's grown in a zero gravity. How does the colony react? Does it grow different than it does down here?
I tried really hard to research that. I really did. And I know that NASA's growing yeast in zero gravity, and I couldn't find any results. And what I was able to find suggests that yes, it grows differently in zero gravity, and I spent like a couple hours on this and I couldn't find the specifics on how, but I think it is different.
I was just going to say the light thing with beer in particular is it has a reaction with the acids from the hops, and that's what makes skunky beer. That's why beer comes in brown bottles, is so it doesn't react with that part of the beer.
Sorry.
Hi. The difference between San Francisco yeast and New York yeast, they're the same genetic variety. I know that my yeast here in Atlanta, I got a couple of samples from San Francisco and from Colorado. Those did okay for a while, but I had a couple of failures, and I grew some from my, set my bowl of stuff out in the backyard for a day, and that grew up and has grown up a nice batch for the last few months. How long, or how many generational cycles does it take if I take San Francisco yeast and bring it down here to Atlanta? Will that then turn into Atlanta yeast?
I think that depends a lot on, is it a closed system that you're not letting anything else into, or are you letting environmental contaminants into it? If you don't let anything into it, it'll breed true pretty much. I mean, it's going to change some as it goes through various mutations, but it's not going to turn into Atlanta yeast specifically. It might turn into something different, but not necessarily Atlanta yeast.
Hi. One year I bought unpasteurized apple cider and it fermented naturally in my fridge and was fabulous. And I could keep it going for a couple of months, but eventually you can't get apple cider anymore. Would there be a way to preserve that yeast for the 6-8 months between unfiltered apple cider, unpasteurized apple cider being available?
I'm sure there is. I don't know if it's possible with home equipment.
Yeah.
I mean, what I'd probably try, and I'm not like, if I were screwing around in my kitchen at home, but I'd write it down so it would be science. Thank you, Adam Savage. If I were trying stuff at home, I would take some pasteurized apple cider that I just grab off the shelf and mix it and take a little bit of that and put in some of my unpasteurized stuff and let it colonize that and see if you could build your own culture that way. It might not work. I'd sterilize the glassware first and whatever, and it might fail miserably, but it's not a big risk factor there. So I would try propagating it and see if it works.
I've never had one as good again.
Yeah.
To address the point of the yeast changing over time, so there's a guy who has worked with archaeologists and has extracted yeast from ancient Egyptian pottery and made bread cultures out of them, and he keeps them pure, but he has to autoclave all his ingredients and all his stuff, and it's insane. It takes half an hour to feed his starter every day. Wow. And so it is possible to keep it pure, but most people aren't, and so stuff from the air or from your flour will get in, and it will change and become whatever. But that's kind of what happens with sourdough for the past 2,000 years. So.
Okay. So there are some industrial uses of yeast, not just industrial beer making, but also it has been genetically engineered to produce fun things like insulin. Are you aware of any other applications in this area?
I mean, I know there's some work being done for bioremediation using yeast, but I'm not real clear on the details on it. What's bioremediation? It's using, cleaning up pollutants and things like that.
Thank you.
Yeast is great to make stuff for you. So if you need a protein, you either genetically engineer E. coli to make it for you, or you genetically engineer yeast, Saccharomyces cerevisiae, to make it for you, whichever's going to be best at it, because those things grow like gangbusters, and we're really good in lab at keeping those alive and keeping them functioning. So you just let it do all the work, as opposed to you having to try to deal with protein synthesis. There are machines for that, and some of them are biomachines for that. We're just going to make but that's not, sort of whatever you want it to make. Like you say, maybe insulin, other proteins, and then you just harvest it out with a blender and a centrifuge, literally a blender and a centrifuge, which is just a big washing machine.
Yeah, yeast are pretty happy to take up genes and start making stuff.
So even though I'm terrible at making bread, I've been venting and mead making for over a decade, and I've had a lot of success just brewing on random bits of anything that has sugar in it, that I get from the store. It might have this and that. Although I have noticed that some preservatives obviously just retard the growth to the point where you can't do anything with it. Is there any particular preservatives that that are problematic that I can just avoid right away, and ones that maybe aren't problematic? I don't know.
Not anything specific, I don't think. Like, yeah, certainly I'm sure some of them are definitely are going to be problematic, but not that I know of. Potassium sorbate. Okay, the audience says potassium sorbate.
Yeah, they'll put that in ciders to retard any kind of bacterial growth or yeast growth. So if you're making your own cider, you want to avoid that big time.
All right, avoid that in cider. Cool.
Or even just salt. I mean, historically.
Have y'all seen what happens when the youngster working at the pizza place forgets to salt the dough and they have to throw away the dough? But if you don't salt one of the points of having salt in your bread dough is, of course, flavor because it's delicious. But one of the other points is that eventually you need the yeast to stop functioning. And so you can find these beautiful pictures of somebody who forgot to salt the dough on a big batch scale. And then they put it in the dumpster, and the dumpster is hot, and it's an oven, and you get the entire dumpster overflowing with baking bread. Just like, bleh, bleh. What do I do?
Yeah, I've seen that.
So we've talked a little bit about yeast cultured in space. I was wondering about the opposite. Has there been any yeast cultivation attempts tried in our oceanic depths?
I don't know. Do you know?
I think most of the extremophiles down there are bacteria.
Yeah.
So there's lots of halophile bacteria that love really, really salty environments. So like the Dead Sea and places like that, it's not dead. You can find bacteria that are really good at living in that one specific niche. But I think it's mostly just bacteria. I don't think you get yeast. I don't think.
I think it's worth noting the reason why they've grown yeast in space is because they're using it as a way of understanding how long-term periods of time in space affect organisms that have DNA. And so I think a lot of the reason it's been grown in space is for research purposes. And so if anyone's grown it in the ocean, I would wonder, like, maybe it would be for research purposes that I don't know about because I don't do water, I do space.
Yeah, just for the purposes of seeing what cosmic radiation would do to its genetics.
Yeah, in fact, they're launching yeast into deep space later this year to understand the effects of long-term space travel on DNA.
The only thing I can think about putting any single-celled organism in microgravity to see what happens. Yeasts do everything by membrane diffusion, right? There's no.
I'm gonna look at the biologists on that one.
There's no structure that lets materials in and out. It's just stuff diffusing through the cell wall.
I think yeasts do. I'm pretty sure they have
Okay.
active transport mechanisms.
Okay.
Because they're eukaryotes. They're more complicated than like bacteria, which might be more like that.
But it occurs to me that some organisms that it emits some chemical in solution. Everything on Earth evolved in gravity, right? So you would expect that if some form of alcohol came out of yeast that was, or some chemical came out of something that was lighter than water, it would tend to convect up and away from it, whereas in microgravity it would just sort of build up around it, which might affect the ability of nutrients to get. That's about the only other thing I think of to be worth trying to mess around with yeast in microgravity for.
And yeasts don't have any way to move besides making more yeasts and
Right.
baking them in a general direction. They don't have motility.
Howdy. I'm making mixed culture beverages at home, mostly beer, also kombucha, where you're primarily trying to get the Saccharomyces, but there's also the acetic and the lactic acid bacteria you're getting. And I have great success with buying commercial products, getting these from the bottom, pitching it in. Tastes great. But it's not very predictable. Is there any way in a home environment I can identify the proportions or ratios of those bacterias so I can understand what my final product will taste like?
How do you feel about buying agar plates?
Yeah.
I'm not really sure what you could do besides actually culturing it and
Yeah.
doing assays.
Whatever you get out of that is like, do you really know what's in that? Like, yeah, okay, this brand probably works, seems to work okay, but you don't know for sure what's in there or what ratios are present. So, yeah, depends on how much science you want to do at home.
Yeah, about the space radiation. Do we have to picture like a petri dish outside the spacecraft because I assume the spacecraft is shielded?
That's a good question.
I can speak to that some.
I think they've maybe done both, but I don't know if you know.
But ISS orbit is about 300 miles. It's not great for cosmic radiation shielding just from Van Allen belts. It does have enough plastic in its walls to also provide some shielding. Generally, they don't leave anybody up in the ISS so long that's going to be a problem. But it is a higher cosmic radiation flux than you're going to get on the ground. So if you want to, if you want to study what interplanetary space would be like, just near a star like our sun, being 300 miles up in the ISS it's not quite as far out there. It won't get as much cosmic radiation from the sun. It may get it from other sources besides the sun, but it's a good place to run an experiment just to see what it would be like.
And a lot of the reason they're doing it is to understand what the effect is on the humans living on the ISS, and so they're going to want the yeast to have similar shielding to what's on the ISS. And even for the yeast that they're sending into space, they're sending into deep space, they're sending in a satellite, which I imagine is going to have probably similar to what they want to put on spacecraft that they might send astronauts out in.
I have read that some ISS astronauts will even, in the little booths where they sleep, they'll surround themselves with bags of water. Water is excellent cosmic ray shielding.
Do you have the professor's pancake recipe?
I do. I have a Google Doc that contains not only the recipe, but the backstory of it. And it actually gave me a bunch of other recipes, including the best sourdough cornbread you have ever tasted. The other, besides the sourdough, the other secret is that you preheat your skillet with bacon grease in it, and then you dump it in, it flash fries this bacon crust around the outside. It's magic. So again, if you email me, TERESHKOVA2001 I will send you those recipes, and also mail you out some starter if you want to ask for that. I have a few cards, or you can track me down after the panel. TERESHKOVA, like Valentina Tereshkova, the first woman in space, 2001 @gmail. It's not going to be right away because but.
But before the question I have, it's not really a recipe. It's pretty lazy. But as a byproduct, like every time I feed my starter, I would dump half out into a pan with olive oil in it, and just let that cook. And then I put everything seasoning on it, and it's like a really nice, really sour pancake, but it's really good. Forgive the ignorance. I'm neither a biologist or a geneticist, but when we were talking about how long until the cultures you're using or the starter you're using is no longer the same starter that it was, I heard a study last year from a geneticist that was talking about how when we look back to our ancestors, though we share genealogy, we don't share genetics. You can go so far back to where you share zero genetics with your ancestors, even though you technically came from them. Is it similar or completely different when it comes to yeasts or other organisms that are constantly reproducing?
I'd say yeasts are probably going to be more similar to their ancestors because of that asexual reproduction than humans are, because humans are constantly recombining their DNA and changing it and everything, whereas yeasts, if they're happy and things are going great, they're going to be reproducing, cloning themselves just again and again. So they're going to be more similar.
Is there any sort of diseases that yeast can get that might like affect their growth or cause your colony to completely collapse, that sort of thing?
You're faster than me on that. You didn't. So, do they get a yeast infection?
If there was, my guess would be it would probably be some kind of virus, but I'm not familiar with any. I think it's more likely that they just experience competition that kills them. This is just spitballing, though.
You've got something else that happens to be growing in your culture that is just, more better at doing the thing. I'm a trained scientist, can you tell? Then it would outcompete. But like on a smaller scale, like these really single-celled organisms, they don't, like, disease isn't really a thing that happens on that level, as to my understanding anyway.
Cool. We got time for about one more question.
Yeah, so obviously we've heard one example of a very special yeast strain that's been passed down for a few generations. But for anyone else on the panel, is there any other type of unique yeast that you've found that you've used in cooking or brewing or what have you? Where did you find them?
I didn't personally find it, but the book I recommended, Alaska Sourdough, the person who wrote it talks about, I guess in the part of Alaska she lives in, they have many multi-generational sourdough cultures. And so, like, I've read about it, and I thought that was really cool, that apparently there's a huge culture around sourdough, and I forget what part of Alaska it is, where basically everybody has a sourdough culture that's a few hundred years old. I know. If you couldn't hear that, there's apparently a sourdough museum somewhere, so that's your homework. Go find the sourdough museum.
And if you want to talk about yeasts that are passed down from generation to generation, Candida Albicans, you got it from your parents.
Give it to your children.
Yeah.
Apparently there are viruses that do infect yeast. Rhinoviruses?
Rheoviruses? Rheoviruses. Okay, cool. How long can yeast live?
That's a complicated question because it's like one individual yeast organism or a spore of yeast that's been preserved, or a colony of yeast as a not quite immortal thing. Like, I don't know the answer to any of those questions except at minimum 130 years old, because that's how old my sourdough culture is. And Jen was saying a few hundred years. So you can keep a culture going for a very long time, but is that one yeast? Like, it's a generation of yeast. Yeah.
But in general, yeast can, at least Saccharomyces cerevisiae, it can double every 1 to 2 hours. So in a couple hours, you're going to have an entirely new set of yeast, but also the same yeast that you started with.
We have no more questions. It's been wonderful having you. We're out of time. Thank you for coming. It's really exciting to get this big a crowd in this room. Thank you so much. Thank you very much. I hope Hope you come back to experience the other Science Track panels. And I was telling Paul McCartney yesterday, God, I said I wasn't going to do that. Thank you for coming. I appreciate it.